Hi, so I'm Kevin Drew. I'm an assistant professor at the University of Illinois at Chicago, and I'm very excited to be here today and to share my work on the hu.MAP 2.0, the global compendium of human multi-protein assemblies. And I wanted to thank the organizers for giving me the chance to tell you about my work today. So proteins are the functional unit of the cell, but we also know that they don't work alone. They work in concert with other biomolecules in the cell. So we can look at essential functions of the cell, such as replication, translation, transcription, and degradation. When we look at the proteins that are carrying out these functions, we say that they form these large macromolecular protein complexes. So we see RNA pull in the mediator complex, we see MCM in the ORC complex, we see the ribosome, we see the proteasome. So many different subunits coming together to carry out these essential functions of the cell. We know that this is a general principle of biology, and it's not exclusive to just essential functions of the cell, but it's really, again, a general principle, and we see this across many different biological functions. So many different questions that I have in my lab are centered around these questions about large macromolecular assemblies. So what are all the macromolecular assemblies in the cell? How stable and dynamic are these assemblies? How do these assemblies function? And what happens when these assemblies break? So if we disrupt these assemblies, what kind of diseases are causes for these? So today I'm just going to talk about this first question. What are all these macromolecular assemblies in the cell? I like to think about this as building a large molecular map for the cell, something akin to, say, Google Maps for the cell, where we can take this very broad view of what the cell looks like and be able to zoom in on different regional views, and then be able to even zoom in even further on, say, a specific biological entity, and then even be able to kind of take our little person and be able to kind of drop it down onto a street level view and be able to walk around and get some biological intuition in terms of the function of these biological entities. So this is the dream. This is the grand goal of what we want to achieve, is this kind of Google Maps for the cell. But to achieve this, we need to answer some basic fundamental questions. And in particular is just what are all the components of these biological entities? And in particular, what are all these proteins that are making up these protein complexes, these entities? Okay, so we need some tools to ask this question. And in particular, a very powerful approach to ask what are the components of these complexes is something called cofractionation mass spectrometry. So here we're taking cellular lysate and we're going to biochemically fractionate this lysate using a biochemical separation technique. So one example is size exclusion chromatography. So we take cellular lysate, we're going to take all the complexes in that cellular lysate and we're going to separate it based on size, where large molecular complexes elute in high molecular weight fractions, medium complexes elute somewhere in the middle, and smaller things elute somewhere at the end. And then we use mass spectrometry to identify all the proteins in each one of the fractions along this biochemical gradient. What that allows us to do then is, for every protein we identify, we can then build and observe an elution profile. So here, there's a red protein. We see that it elutes at this high molecular weight fraction, and then we don't see it again. And then these other proteins, this orange protein and yellow protein, also have very similar behavior. They elute at this high molecular weight fraction, and then essentially we don't see them again. So we can observe that these three proteins co-elute together. And we see that this blue protein and this other blue protein and green protein all have similar behaviors to each other that are distinct from this red, orange, and yellow protein. Okay? And what we can do then is build a computational pipeline that identifies these co-eluting proteins and identifies them as protein complexes. Okay? So this is a very powerful approach in which to statistically identify protein complexes. And what's also very powerful about this is there's no antibodies or genetic tags required. And so you can apply this to both non-model and model organisms. So we've applied this to human and mouse and frog and flies and sea urchins and sea anemones, as well as many different plant species and other organisms. Okay. So we've applied this to thousands. We've done thousands of fractionation experiments and identified tens of thousands of protein interactions. But we're not the only ones that are interested in this in discovering protein complexes. There's other groups that are doing affinity purification mass spectrometry experiments and identifying tens of thousands of interactions with this powerful approach. And then there's also proximity labeling experiments that are becoming more and more high throughput. And then there's other high throughput proteomics experiments that we can apply statistical techniques and identify protein interactions in that way. So in total, we have over 15,000 mass spectrometry experiments from different cell types, different tissues, and different parts of the proteome. And so these are really orthogonal approaches. And I took the opportunity to be able to integrate all of these data to build a more complete and accurate set of protein complexes. So to do that, I built a machine learning framework, and I don't have time to talk about all the details here, but essentially what I've done is I've taken all of the individual data sets here, and I've reanalyzed some of them using different statistical techniques. I've then built a machine learning classifier to identify individual protein interactions. I've been able to then build a protein interaction network. I've then used unsupervised clustering techniques to identify dense regions within the network to identify protein complexes. And then the final set of protein complexes is something I call hu.MAP 2.0. So there's a number of different ways we can evaluate how well the map, the protein complex map, is performing. And one way is in something called a precision recall analysis. So here we essentially have a leave-out set of protein complexes, and we're evaluating essentially on the x-axis is recall or true literature curated complexes, and then on the y-axis is essentially precision. A perfect precision recall curve is essentially a straight line across the top. We're 100% precise over 100% of the benchmark. And what we see here is 3 individual data sets by themselves before we've integrated them. And we see that they actually do quite well in their high confidence region. They're very precise. But as we increase in recall, they quickly drop off in precision. We can integrate all of these data together and we push this curve up and to the right. We're becoming more accurate and more comprehensive. The other thing that we do once we develop our map is to go searching for things that we expect to see. So, so this is sort of the fun part. We get to go looking for some known biology, go looking for some positive controls to make sure that we're generating some things, some complexes that make sense in terms of biology. We expect to see things like the proteasome. We expect to see things like the ribosome and the mediator complex. And so when we go looking, we see the 19S base lid of the proteasome, we see the 20S core of the proteasome, we see the mitochondrial ribosome, we see the mediator complex. So this is essentially some comfort that our pipeline is generating some novel biology. So this is very good news. Okay, so our previous map, one thing that's really quite powerful in our that we've done in our previous maps is gone looking for essentially a guilt by association to identify candidate disease genes. And here is just an example of that, of looking for ciliopathy disease genes. This is where we've recapitulated a lot of ciliopathy ciliary biology, where many of these are ciliary disease genes, and we've identified them into complexes, known complexes like the BBsome and the IFDB complex, the IFDA complex, and others. But what was interesting about this is we identified some genes that were physically associated with these known ciliary complexes that were not involved in ciliary biology previously. So these proteins in green here weren't previously associated with ciliary biology. So these provide hypotheses as to their function with ciliary. ciliary biology. We followed up on a number of these, but one particular one that we followed up a little bit more closely was ANCAR D55. You can see over here that we localized ANCAR D55 to cilia, so this was predicted by our MAP, and then we also showed that ANCAR D55 was necessary for development, which phenocopied the IFTB members. Okay, so we thought about this a little bit further. In terms of looking at understudied proteins and highly uncharacterized proteins. So this is a bit of a hot topic, and we're really excited about Georg Kröscher's and Yuri Rapsipler's recent comment in Nature Methods, where they dig down a little bit deeper into some of the reasons why some proteins are just highly unstudied compared to other very well-known studied proteins. And so I'd encourage you to kind of take a look at some of those reasons as to why. But one really powerful approach to studying highly uncharacterized proteins is to use unbiased methods like our hu.MAP 2. And so we've looked at over 4,000 highly uncharacterized proteins in the human proteome and gone searching for them in our protein complex maps to see if we can't transfer an annotation based on their physical associations. So one particular example of this is a protein called C7orf26. It's a highly uncharacterized protein, very little, hardly anything is known about it. And C7orf26 we've identified as part of the integrator complex. So the integrator complex is involved in transcriptional regulation. What we see here is that it's co-eluting with C7orf26 is co-eluting with the integrator subunits. So this is ion exchange cofractionation data. And here on the x-axis is fractions. And we see individual components here all co-eluting here in HEC293 cells. We see that this is conserved in fly. So again, fractions on the x-axis and all of these subunits here from the integrator complex and C7orf26 are all co-eluting. So this is pretty good evidence that C7orf26 is part of the integrator complex. We can also look in affinity purification mass spectrometry data where integrator subunits are being pulled down. And we see that C7orf26 is being pulled down with it. So this is all based on mass spectrometry data, and we've identified, we can then transfer the annotation to C7orf26. Recently, we were excited to see that Jonathan Weissman's lab has used an orthogonal approach with his genotype-phenotype screens and his Perturb-seq framework to identify that C7orf26 also is associated in his screens with the integrator complex. So now we're seeing essentially transfer of annotations based on multiple high throughput assays. So this was exciting for us to see our prediction and our annotation validated. Okay, so in total, we've been able to annotate over 270 uncharacterized proteins. So this is the transfer of annotation from an uncharacterized protein based on its association with a known complex. And you can see some of these examples here where we have the ribonuclease H2 complex associated with a number of other highly uncharacterized proteins, an exon-exon junction complex to CCDC9, and a number of other examples that are providing testable hypotheses in which we can go into the lab and test. So we want this to be a resource for people to use, and so you can go to hu.MAP2.proteincomplexes.org. You can search a gene of interest here with our web interface. This is a cytoscape view of all 7,000 protein complexes. We cover about half of the human proteome, about 10,000 unique proteins, 57,000 unique interactions, and again, we cover about 270 highly uncharacterized proteins that we've been able to transfer an annotation. This work was recently published in Molecular Systems Biology, so you can see more of the details there. And again, this is a resource for the community to use, and so we're really excited to see people starting to use it. One example that we found in the literature, we're really excited, is from Arnie Ellefson's lab, where he's using hu.MAP2 as the foundation for his proteome-wide structure determination study, where he's using AlphaFold 2 to determine to predict the structures of many complexes, and using hu.MAP2 as a basis for that. So we're really excited about how hu.MAP2 is being used in the community. So with that, I just wanted to thank my 2 postdoc labs, the Marcotte lab and the Wallingford lab, and the NICHD for funding. And then I recently started my lab at University of Illinois, Chicago, and so I have positions open for postdocs and students. And so if you're interested in protein complexes and identifying them and characterizing them and modulating them, I'd be happy to talk. And so thank you for your attention. Yeah, I look forward to hearing from you.